Seriously, I hit that record button. Did anybody see me hit that record button? That's unfortunate. That's what that is. Okay, so Kristen, I did not do study guides, and here's why. Because study guides make my thinking become your thinking. And so that's where the concept maps become really, really, really tools that you guys should start to utilize. So ideally, students who tend to be most successful in this course study a little bit at a time. Now, I know you've heard this. This is something you've heard for a long time, I would assume. And it's because it's true, right? Doing a concept map for each chapter becomes your study guide. There's also study guides within the study areas on Mastering Biology. So if you go into our Mastering Biology study area tools, there's all sorts of resources. And so I don't do study guides because it's just me thinking for you. And then everybody, quite honestly, everybody gets all ticked off when it's not perfectly matched. And I tend to ask really big global questions on study guides, and then there's not one precise answer, and then that makes you upset too. So if we go to carbon, there's all sorts of stuff you can go through in here. There's key terms, there's practice exams, there's all sorts of stuff you can do. And so that's what I would utilize more is concept mapping and using your Pearson resources that you've paid for. More questions? Yes. So it was just asked, quizzes are limited to what I present to you, or exams limited to what I present to you? Yes. I got a tickle, sorry. So no, it doesn't matter if it's asked 1,000 times on a practice exam on the study guides in Pearson. If I've never talked about it, I will never ask you about it, ever. Never, ever, ever. Yep, Amanda, the exam will cover everything you learned up to the point of the exam. We'll never start new material. Whatever is done on Thursday will all be included on the exam. Okay, so let's then continue with what will be on the exam. We were talking about lipids, and we were talking about how we made our triglycerides. So remember, triglycerides are a group of lipids that are together because they, sorry, let me say that again. Triglycerides are a specific type of lipid. Lipids are in a group together because they are hydrophobic molecules. They are a largely diverse group. They're a big group of molecules. They don't have small little monomers that are their little building blocks. Instead, they're hydrophobic, and they're hydrophobic because of these long carbon chains, right? You have these long hydrocarbon chains that makes them hydrophobic. They are nonpolar. They're covalently nonpolar bonded. Now, we were looking at this ester linkage. So let's start there and draw it out so that we have it nice and slow, okay? So there is no help for a pen, I found out, so you always get my finger. And so, you know, there we go. So let's draw that glycerol molecule. We have a carbon that's got 3 little carbons in it, or a carbon molecule that has 3 carbons as its core. It's got a hydrogen here, hydrogen, hydrogen, hydrogen. And then it's got these hydroxyl groups. There's 3 of them, which sets up the triglyceride part. So this is that glycerol molecule. Okay, that's the glycerol molecule. I think I spelled that right. I cheated to make sure I put the chemical symbols correctly, but I didn't do my spelling. So you have a long fatty acid chain. And so the long fatty acid chains are going to attach at each one of these hydroxyls. And it's going to do so via a dehydration reaction that's going to result in a covalent bond that we're going to call an ester linkage. An ester linkage is between a hydroxyl and a carboxyl group. Sorry, I don't know why my child's calling me. So we need to look at those two functional groups. So when we look at our fatty acid chains, we're going to have a carboxyl group over here. And then we're going to have that big hydrocarbon chain. And I'm not going to draw all the carbons and hydrogens because there's no need to, right? We'll draw that kind of abstract view of it. Yeah, Dylan, what's up? Oh, yes. Thank you. There should be a hydrogen coming off of that third carbon right here because those valence shells were not complete. Thank you. It was an unhappy, unstable molecule. This is why my biology grades were always slightly better than my chemistry grades. So, oh shoot, and I didn't draw my carboxyl right either, did I? So this is an OH. Okay, so what we have here is a carboxyl group. Let me write that. Like I spelled that right. And this is our hydroxyl group. The ester linkage is going to be between the two of them. Now keep in mind this is going to happen in each of our fatty acids. So what happens is you have to have two hydrogens and an oxygen leave so that you have the water that gets squeezed out. So that's what you have right here. These, oops, sorry. These are going to squeeze out. And then you're going to have your ester linkage that will result. I can't draw that with the space I have. I'll go back to the other picture so you can see what it looks like. This, for sake of labeling, is a fatty acid chain. Oh, and there's no way to increase that font. Fell down the rabbit hole of that for a long time before I figured that out. So the result of this dehydration reaction will be a triglyceride, and each one of those fatty acids are linked to that glycerol via an ester linkage. The ester linkage happens between a hydroxyl group on the glycerol and a carboxyl group on the fatty acid. That's kind of the level in which you want to be comfortable with. So if we go back to this picture, that's what you're seeing right here, nice and neater than I drew it, right? So you have your glycerol, you have your fatty acid, the dehydration reaction squeezes out that water, covalently bonds these guys together. The resulting bond is called an ester linkage, and that's between that hydroxyl group and that carboxyl group. And that happens at each one of those hydroxyl groups on that glycerol. That's why it's called a triglyceride. It's got three fatty acids on a glycerol molecule. And then you have these huge hydrophobic regions, which makes fats hydrophobic. So let's look at saturated versus unsaturated. And so what fat are you supposed to eat? Does anybody know? I know there's a lot of exercise science people. A lot of people want to be, nutritionists from the course. So which one are you supposed to eat? The saturated fats or the unsaturated fat? What's the healthy one for your heart? Anybody know? Yeah. Yeah, unsaturated. Yep, unsaturated. So the unsaturated fats are healthier. And so that's these guys. So what makes them unsaturated? So the unsaturated fat, and let me kind of highlight which ones I'm talking about specifically. This one's unsaturated, this one's unsaturated, this one is, and this one is. Okay? They are different because they're bent. Do you see how they're kinked? That's different than these saturated ones that are completely straight up and down. So saturation has to do with their hydrogen bonds. If you are a saturated fat, what the saturated word means in terms of chemistry is that every one of carbon's valence electron needs is being filled by a hydrogen. It is saturated with hydrogen. If it is unsaturated, it's not saturated with hydrogen, it's unsaturated with hydrogen. So something else is helping to fill carbon's valence electron shell. And in the case of a largely carbon and hydrogen molecule, it's going to be another carbon. So these little kinks that you see, these are double bonds. These are carbons double bonded to another carbon, which changes the orientation of the bond direction, and so it changes that shape of that particular fatty acid. It's still largely carbon and hydrogen. It just now is kinked because of that double bond. And let me show you a couple pictures. So right here are two really helpful pictures that show it a little less abstractly. So if we look first at the saturated fats, right, that's my favorite fats, my butter that I love. I love the butter. This is what the fatty acid chains look like on a saturated fat. You have your glycerol molecule. This is our ester linkage right there. And each one of our fatty acid chains is completely saturated with hydrogens. That means that every carbon is valence shell is being filled by those hydrogen electrons, right? So it's a complete valence shell. This, I guess, another way to say it, the definition from the book is the maximum number of hydrogen atoms and no double bonds. Most animal fats are saturated. So that's most. That doesn't mean all. So the result, because of the saturation, is that these can be, here I'll zoom out a little bit, these can be then tightly packed, right? And so at room temperature, these remain solid. So saturated fats at room temperature remain solid. So think of all the fats that you deal with, and the ones that are solid at room temperature, those would typically be saturated fats. Why are they a little worse for you? We are not too much, you know, our body stays at standard temperature. So when we eat them, it's not really warm enough to melt this necessarily in our body. And so they stay tightly packed in our body. And so if you eat a lot of butter, that fat, and as it's stored, becomes tightly packed within our body. And so it can end up clogging our systems, right? We have capillary systems, arteries, veins. Stuff needs to move through that freely. Because these are hydrophobic, they're not going to dissolve. They're not solutes that are going to dissolve. They're non-polar bonds. They can't break apart those hydrogen bonds between water molecules in our body. So they just kind of stay together. And so they clump and clog things. And so that's why saturated fats over time are not great for you, right? They're not the best thing to have in excess. Are they fine to have a little bit of? Sure. So then if we look at unsaturated fats, those are going to be things that are liquid at room temperature, and that's typically a quick way to kind of distinguish them. Oops, I lost my text box. It's usually a quick way to distinguish it. It's not a surefire way to distinguish the two fats by any means. So your grapeseed oils are really good. Unsaturated fats, olive oil is obviously a really great unsaturated fat, probably the most commonly known one. And so what makes them different is they are not saturated with the maximum number of hydrogens. They're going to have at least one double bond. Now remember, we present the rules, not the exceptions. So each kind of specific fat is going to have its specific mix of fatty acid chains. So again, you have your glycerol molecule right there. This is still an ester linkage. This is still a fatty acid chain that's a hydrocarbon. It's still hydrophobic due to its non-polar covalent bonds. All of that is still the same. The thing that's different is that you have a double bond. What's the double bond between? It's going to be between two carbon molecules. And so the carbon molecules use each other's valence electrons, two separate carbon atoms, and they're going to share those electrons, and it causes a kink. It actually causes a cis double bond, so the double bond's on the same side, and so it causes a particular kink. And so typically plant fats and fish fats are unsaturated. That's typical. So that's not all of them. These are better for you because they don't tightly pack. Because of that kinking, they still allow space for stuff to move around. They don't stay like in a solid butter state in your body. So it's amazing how much of our language is really informed by chemistry, but we have no idea. These make for great cooking oils, unsaturated fats. A lot of them have a high heat tolerance, and so they're really great to cook with. Typically, we would talk about this, but so if you have a grease fire, why can't you just throw water on a grease fire? So if I put a bunch of oil and then, oops, I lit it on fire, the oil in my pan, why can't I put water on that? Why is that bad? Yep, oil and water don't mix. They're hydrophobic. So when you have hot oil and the water and you push it together, it's like my little bottle on extreme heat. And so they just expel each other and it makes the fire bigger. And so if you have a grease fire, that's why you want to put baking soda or flour on it, because it's really working within the chemical guidelines that we have. So, I want to talk a little bit about a fat that a lot of us maybe know about or try to avoid. I will never ask you about this. This is just to kind of inform you how chemistry informs our life. So trans fatty acid. Trans fatty acid, what we do is we take the cis bond and we change it to a trans bond. Okay, so we take the cis bond, where that double bond would be on the same side, causing the kink to go on the same side. And what we do is we make it a trans double bond. So that's the wonderful stuff about science in general. This is really an example of engineering, trans fatty acids. And it's in a lot of our foods, and we'll talk about that in a minute. But, you know, we understand biological principles, like vaccination development is a classic example of this. Scientists, in of their core nature, aren't making anything. So scientists don't have a goal to make a vaccination. But when you're making a vaccination, you're basing it upon these hypotheses that have been unable to be falsified of how our immune system works, right? So trans fatty acid is the same thing. A lot of our food industry is based upon these biological, chemical, physical principles, and then we manipulate it, right? And so that's not really doing science, it's doing engineering, but science is at its core. So trans fatty acids, they're bad for you, right? When I had kids 15 years ago, it was a big thing. You were always trying to look at packages. Oh, is there trans fatty acid in this? I can't feed this to my baby. This is not good, right? And why? We were looking for partially hydrogenated oil. That was what you were always looking for in your ingredient list. And so hydrogenation is actually the process of converting unsaturated fats into saturated fats. What they do is they make the natural bend go straight by adding the hydrogens back in. And so it's just manipulating. It's taking that double bond that should be a cis orientation, so on the same side, they add some hydrogens to it, it bends the other way, so it's on the trans side now. And now what you get, what you get is a trans double bond, but what you've gone from is an unsaturated fat that's bent, and you have a trans fat which looks straight. It looks like my saturated fat over here, but it's not my saturated fat. Okay, so even though it looks like it, it's not exactly the same. So we should sort of know now, if it's not exactly the same, it's going to function slightly different. So that's the case here. So when we hydrogenate vegetable oils, we're taking unsaturated and making them saturated with hydrogen, thus changing their double bonds from cis to trans. Why are they bad for you? They are exceptionally good at clogging your body. Because we've manipulated them, they just stay together even better than what a natural saturated fat would be. So if you had your dietary choice, you should eat a naturally saturated fat over a unnaturally saturated trans fat. Does that make sense? Your body will be happier with this in the long run than this. This is very bad. That's why now it's harder to find as an ingredient in food. It's really been kind of moved away. Why do we need trans fat in our food? What would it help to provide? What benefit does this provide in our food sources? It's in a lot of cookies and baked goods and crackers and chippies. It's a preservative, right? We got to have food sit on a shelf for a long time. If it gets really warm, we can't have the butter melting, right? It's got to stay shelf stable. Trans fatty acid does that really well. So, and also, unsaturated fats can sometimes be cheaper to come by. I said most fats were animal fats. So a plant fat, like a vegetable oil, is really cheap to make. And then all you got to do is take it from this to make it into this, and now it's shelf stable. Now we've taken a liquid oil, we've made it a solid, and voila, my cookies stay on the shelf for a year. So it's a great preservative. Okay, it also makes shipping oil to like a McDonald's easier, because you can ship it in a solid form, and then they can heat it up and it fries real great. You can't, it's harder to ship a liquid. It's more tricky, right, logistically. So there's a lot of benefits. So just some food for thought, I guess. I won't ask you about it, but it's kind of interesting to know how much, chemistry is in your food. Questions on that? Don't eat trans fat, it's bad for you. Okay, so let's move on to our next group of lipids. This group of lipids we are going to look at extensively, like so much. We're going to have a whole chapter on these bad boys. So I'm going to kind of go through it relatively quickly. In our cell structure, we're going to look at them in a ton of detail as well. So notice, this is one thing I said to my other section of 120 lecture, right? Because I've got 2 sections. Most of your professors teach multiple things. Just keep that in mind, right? So the more information when you email us, the better. So if you think about it, the glycerol molecule was this color. The fatty acid chains were that color. Now we've got a new color here. So we've got an alcohol. We know we're going to have a hydroxyl group on that. Why do we know that? Well, because I just told you. And OH is, there's going to be that hydroxyl group of OH makes things in alcohol. And then notice right here we have a phosphate group, okay? Phospholipid. Phosphate group. So this is technically called the glycerol phospholipids. No one calls it that. Everybody calls it a phospholipid. Because who wants to say that big old mouthful all the time? Nobody. So we still have a glycerol molecule, so that's similar to our fatty acids, to our triglycerides. But now we have a phosphate group. This is a dehydration reaction that results in the ester linkage, same thing. This is still going to be a dehydration reaction, but we don't need to know any linkage names there. Woo! Remember, an alcohol is going to be a polar molecule. It has that hydroxyl group in it. Hydroxyl has an oxygen in it. And oxygen is going to have electronegativity. So this changes how this will chemically behave a little bit. However, it still has these large hydrophobic regions, which still categorizes it as a lipid. So keep that in mind. That's why we don't have my little Legos out and our little building blocks. It's because lipids have a regular pattern. I'm going to read that question again. Is the phospholipid the PO4 in the alcohol? Oh, no, the phospholipid is, I guess, the phospholipid's the whole thing. The reason why it has phospho in it is because of this particular chain hanging off of the glycerol. But the phospholipid is the whole structure. Let's look at the next picture. I think that helps. So here's the picture of it, like actually not with, boxes. So the phospholipid structure is your membrane. Phospholipid bilayer, what we're talking about, guys, this is your cell membrane. Oops, sorry, I don't want it there. This is your cell membrane. Okay? That's the plasma membrane. That's the cell membrane. It's the phospholipid bilayer. We call it a lot of different things. We're going to have a whole chapter on it. So we have a hydrophilic head, okay, and we have these hydrophobic tails. So notice, because of the chemistry, we have a unique shape. So this picture does not really do it justice on its shape. It tells you what the parts are, but it doesn't tell you how it actually ends up being chemically. And so if you look here, what we end up having is here is the glycerol molecule, that original part of the glycerol right here. See how it's labeled? Then you have the phosphate group, and then you've got the alcohol group up here, which is a little cut off, okay? You have the hydrophobic tails, and oftentimes we have a mix of saturated and unsaturated fatty acid chains in our membranes. We'll get into why you would have a mix, but just know that it's not going to be completely all unsaturated or completely saturated or completely unsaturated. There'll be a mix. Biggest thing that you need to know right now is that you have a glycerol molecule with the 2 fatty acid chains, which results in these large hydrophobic regions. And because you have a phosphate and an alcohol group, you have a large hydrophilic region. So this hydrophilic head loves water. The hydrophobic tail hates water. And your cell membrane is in a bilayer. Bi means 2. Bi always means 2. And so that means if you kind of can, I'm going to zoom in for you so that I'm going to force you to look at it. If we zoom in, you can see that right here, we have heads. And then right here, we have the heads. And in the middle are the tails. Now, your book likes to call it a sandwich. And sometimes the sandwich analogy makes super sense, and sometimes it just doesn't. So I would say, the peanut butter and jelly middle is the tails right here. And then the heads are the bread. So the heads, if they're hydrophilic, are they polar or nonpolar? If the heads are hydrophilic, are they polar or nonpolar? They're polar, right? Yep, I love it. I love the different responses. That's perfect. So that's something that you want to keep straight. I cannot get that box where I want it. Polar, perfect. The heads are polar. The tails are hydrophobic, so are they polar or nonpolar? Nonpolar. They do not have an affinity for water. The heads have an affinity for water. Because it's in a bilayer, and because of how this chemistry is set up, your cell membranes just assemble randomly based upon the heads wanting to touch the water. So in here, this would be the cytoplasm, which is largely water, and out here would be the, what do I want to call it? Let's just say the aqueous, the water solution around cells. I don't want to add too many terms that are not necessary to know, and solution spelled wrong. There you go. Okay? So because of those polar heads and them loving some water, they are going to touch the water, and those tails, those fatty acid chains, are going to be in the middle. This is going to set up all sorts of things within our cellular environment. It's going to set up the fact that we're going to have helpers to move things in and out of our cell membrane. If you've ever watched any videos on cell membranes, they're often called the bouncers or the barrier for our cells. It's because of this water chemistry that they are. Okay? Our little phospholipids, much more to come on them. We'll kind of even add to this very simplistic picture of them. There's a lot more going on in our membrane than just these. Questions? So the functional groups in this one here, you're still going to have gosh bless America. You're still going to have the carboxyl groups doing a dehydration reaction with the hydroxyl group. That's always going to be the same. Then here you have the phosphate group, which is a group. That's what the phosphate group is. And then up here you're going to have an alcohol group that's going to help to make it. It just makes it charged. I know you don't see an OH up here. It's because it's gone when you bonded here. So it's not necessary for you to know. Sometimes when you guys ask questions, I love it, but I don't want to take you deeper. deeper sometimes helps you make you understand it more, and then sometimes it confuses you more. So I like to lie in that tension of, let's just get us right where we need to go. And then what the beauty is in an education where you take some chem is you use that chem then to add to it and use your bio to add to your chem. That would be ideal. You can make concept maps between classes. Oh, that would be exciting. Okay, so here's kind of a zoomed up picture of it so that you can reference it. Okay, so you have the hydrophilic head, the hydrophobic tails, which if we kind of draw our arrow are right in here, our hydrophilic heads are right in here. And then in either side, there's water, right? Your cytoplasm is largely water. This kind of extracellular material stuff is largely water. And so that's how it sets up the interaction. Oops. That got crazy. So Brooklyn, technically that carboxyl and hydroxyl group, when they're linked, it would be called an ester linkage. Yep, so back there on that picture, right here, this would be an ester linkage, yes. Oh, right here? It just, so the oxygen has a space left and the carbon has a valence electron left, and so it just comes in and it does it via a dehydration reaction too. No, not that we need to know. Awesome bond. Okay, much more on our little phospholipids to come. The last thing are the sterols, or what you could think of as they're called sterols, but steroids would be one. But really, hormones and cholesterol are kind of our two big ones. There's no ester linkage in these guys. Okay, oops, sorry. No ester linkages, because look, they look like little honeycombs. What makes the sterols is that they are 4 fused rings. They're largely going to be hydrocarbons, right? So they're going to be carbon and hydrogen. So this molecule right here, these little honeycombs, each one of those are complete carbon and hydrogen molecules. The first steroid is cholesterol. So this is what your little cholesterol molecule looks like. Here's a hydroxyl group off of a carbon ring. Here's a methyl group off of a carbon ring. Cholesterol is actually something your body needs and it makes. It's a common membrane lipid. So within our membranes, we're going to come to start to revise that initial picture I just showed you. And there's stuff in our membranes that helps to kind of navigate those weird hydrophobic tails and the hydrophilic, because it's kind of weird chemistry in the membrane. So there's other stuff embedded into our membranes, and cholesterol is one of them. So more to come on cholesterol. Too much cholesterol is bad because it's a lipid. And so it's not going to dissolve in water. It's not a solute that dissolves in water. It's hydrophobic. It's a non-polar bond. And so it can build up and clog your highway for blood just like butter can, just like those saturated fats can. The other one, a lot of sterols are hormones. So you have testosterone, progesterone, estrogen, pretty important ones for our bodies, for our primary sex characteristics and our secondary sex characteristics. We'll get into those this semester. Cortisol is another one. So what makes these all the same, right, is that they are 4 fused rings. And then look, they've got all sorts of different functional groups on them, right? So this speaks to that great diversity that carbon provides due to the fact that it's so versatile with its bonding. And because we're largely carbon and hydrogen-based molecules, those functional groups change the function. And there's even a little stereochemistry in here for people who have taken some more advanced chem. You know, there's some things coming at you, coming away from you, these functional groups. So these guys are together because of their 4 fused rings. That's what makes them together. questions on these? We'll start to look at them in context as we go this semester. Okay, so hang on just a second. I thought I had loaded some different stuff into here this semester that I wanted to share with you. And now I'm wondering where, oh, there it is, it's down there. Okay. I'm like, I tried to load in some stuff. Okay, so let us continue. This is our next one. This is a pretty big group. And I don't know how to preview this and then fix this, because when I tried to preview this today so that this wouldn't happen, and I adjusted everything, and now it's still happening. The best laid plans. I don't know why this is happening. So this should say proteins and amino acids. That's what that says. So our next big group are proteins, and proteins are made up of amino acids. We're going to talk about this a lot. So this is our next big group, biological group. We're back to the idea of our monomers and our polymers. So proteins do literally almost everything in our bodies. They are incredibly important. They are the expression of our DNA. So we'll look at proteins in kind of really specific ways. Probably the biggest way we'll look at them is enzymatic activity and how they help mitigate our metabolism. But they're your hair and nails. They are in your blood. Your hemoglobin is a protein. If you have an off hemoglobin, you certainly feel the effects. Brain and nerves, they touch. Enzymatic activity we'll look at a lot. Your muscle proteins, right? When you build muscle, you need proteins to help you do that. How our messages get sent. If it's not a hormone that's sterile, it's going to be a protein message. Antibodies. Okay, the antibodies that are made, they are protein-based. They're responding to the proteins that the viruses are making. So we're going to look at that. And so these guys do everything. Everything I look at that is physical about you is due to a protein. Okay, so we want to look at how do we make these little proteins. Why are you being that nice? Okay, so it's a macromolecule. The monomer is the amino acids. That's their small little building blocks. Okay, so this is the monomer. Just like it says there. You should know that. So what do you need to write down? This. The polymer is called the polypeptide. Now, amino acids are going to have an amino group. And then we're going to look at how they bond between some other functional groups as well. So a polypeptide is many amino acids linked together by what we call peptide bonds. So remember with our carbohydrates, our monomers were monosaccharides, and they linked together via a dehydration reaction that resulted in a covalent bond that was called a glycosidic linkage. And then we just talked about our fatty acids with our phospholipid fatty acids and then our triglyceride fatty acids. They link to that glycerol molecule via a dehydration reaction that results in a covalent bond that's called an ester linkage. Now we have these polypeptides that are going to be many amino acids that are going to link together via a dehydration reaction that's going to result in a covalent bond. And that particular bond is called a peptide bond. So peptide, protein, peptide, polypeptide, the P should get you here, hopefully. And then all you got to really remember is that ester linkage one. So what proteins do, right? They're the most complicated chemicals on the planet, which is kind of interesting. There's 20 different amino acids. So they all are going to have the exact same skeleton that's carbon-based with 2 of the same functional groups. And there's going to be 20 of these kind of like side chains. That's their wild card. If you took math, remember when you could figure out, how many different ways can you put 20 things? And you did those little exclamation mark equations. There's countless ways that these amino acids can link together to make proteins. Proteins are huge structures. Oftentimes, polypeptides link up with other polypeptides to make even bigger proteins. So it's endless what they can do for our body. They keep us alive, right? They regulate digestion. They help defend against invaders. They help you feel emotions. They do everything for us, right? So we're going to, you could do a whole course on just one subset of these particular macromolecules. So we'll look at them as we go and how they relate specifically throughout our chapters. So here's one type of protein. Don't write this down, right? Sometimes this is just for context. So here is the substrate. So this is sucrose. So remember sucrose with that OSE ending, that is a carbohydrate. This is a glycosidic linkage right here, and there's two monosaccharides glycosidically linked together. So sucrose is a disaccharide. That stuff, like as you go through the study, that stuff that you should be doing, and talking it out loud is better than just looking at it passively. So just like with our lactose, we need to break this down to its two smaller parts. And in this case, the two monosaccharides that make up the substrate sucrose are glucose and fructose. So our body's like, I got you. So in your saliva, there is an enzyme. An enzyme is a specific type of protein. It's called an enzymatic protein. And this is called sucrase. So it's very much just like our lactase that was helping us break down our lactose. That lactase was a protein as well. So notice how this is like perfectly shaped. It's like that little sucrose is just perfectly suited for that enzyme. This is all chemically driven. We have to have the right amino acids linked up to make the sucrase so it has the right shape so that it functions correctly. Wrong shape, it's not going to fit the sucrose, it's not going to function. So in your saliva, hydrolysis begins to happen of the sucrose. So it comes in with the help of the enzyme sucrase, and it's going to help to break apart that glycosidic linkage via a hydrolysis reaction. And now you're going to have glucose and fructose going through your digestive tract that are going to be smaller monosaccharides, which then will be able to enter in your cellular environment past your phospholipid bilayer to move towards your mitochondria to be extracted for energy. As you work towards it, that's kind of the idea of concept mapping, right? Trying to connect things and talk your way through it using a lot of the language from other things. So let us look at how this works. So here's the amino acid. I'm going to have to get IT over with me again. Okay, so amino acids, that should say amino. That's our monomer. Here's what it looks like. So let's notice a couple things. This is always going to be the same no matter what the amino acid. So this will always be the same. This little side group, this is what changes. There's 20 different R groups leading to the 20 different amino acids. So in the middle here, we have our centralized carbon. We have a carboxyl group, and then we have the amino group. Remember, on the amino group, the biggest signifying thing is that it has a nitrogen in it. That's why we need our nitrogen-fixing plants, because we need to consume nitrogen to help us with our amino acid building. We make some amino acids, but we need to consume some as well. So always and forever, this is going to be the kind of the carbon skeleton of the amino acids. And then you're going to have these different side chains. The different side chains, if you haven't kind of gone there thinking yet, are going to have different chemistry to them. And they're going to interact differently, which is what's going to cause so many different specific shapes for different proteins. You get the wrong amino acid, you're going to have the wrong shape based on the chemistry. So this is essentially right here, this is essentially one Lego. If you want to take it back to our little Lego idea. So we're going to click together a bunch of Legos via a dehydration bond that's going to then have a covalent bond that results. It's going to be called a peptide bond. And a bunch of amino acids are going to peptide bond together to form the growing polypeptide, many peptide bonds that then will get processed even further in our cell. The bonding is going to happen from the carboxyl to another amino. Okay, I think that's the next slide. Oh, I have the side chains. The good news is that you don't have to know the 20 side chains. There's no reason to know the 20 side chains for this particular course. You will have to know them eventually one day, but you can have non-polar side chains. So one thing I want you to notice is each thing in purple is that carbon kind of skeleton that we're going to find in each and every one of our amino acids. So you have the centralized carbon. This is your amino group. They've now taken it so they're not showing all the bonding. And then here's your carboxyl group. It's the same across. All of these side chains here, these are all nonpolar. So they're not going to interact with water. They're going to be hydrophobic. So that's going to dictate how they are going to end up folding once they're all linked together. Then you have polar side chains, right? Notice, same central kind of purple box. The chemistry of the amino acids is the same. What changes are these little R groups. So now you have all these different little R groups. Notice they do have some sulfur in them. They do have some nitrogen in them. They have some variation, but these ones are all have a polar side chain. So they're hydrophilic, so they're going to interact with water. Then we have kind of ones that become charged based on how they are. And so they're considered hydrophilic because of this, but they're either going to be acidic, which is going to be negatively charged, or basic, which is positively charged. Again, no need to know any of that. Just really notice here that you always have this same skeleton. That same core is always going to be there. So as we go through to do the peptide bond, let's look at how they link together. So here is the peptide bond that is going to bond the new amino acids to the growing polypeptide. So between what two groups is this bonding happening between? It's bonding between the carboxyl and the amino group. So if you look right here, here is a carboxyl group. If you look right here, here is an amino group. Now again, the colorations just don't denote different side chains. You have a dehydration reaction, not hydrolysis, right? Because that's the synthesis. You're squeezing out the water. And what results then is a new peptide bond that now we have a tripeptide bond right here because we have we have 1, 2, 3 amino acids that have been peptide bonded together. Notice again that you have to kind of assume or account for the water that got squeezed out. And remember that this is the carboxyl side and this is the amino side. And that's how that one happens. And it just keeps going and going and going. So how do we know, I'm going to skip ahead just really quick. How do we know what, and I'll go back, I promise you. How do we know what amino acid goes where? It's in the DNA. So within our DNA, we have directions, and the directions, oh, that's not helpful at all. The directions from the DNA are what's going to dictate it. I thought I had the picture. You hate when you think you mess with slides enough and then you haven't. So the DNA dictates what amino acid is going to go there. So in your DNA, remember how we talked about that central dogma the first day of class? So we talked about, yeah, I can. So we talked about the central dogma DNA. Let me just write this really quick. The central dogma is DNA to RNA to protein. This is how it's happening. So your DNA carries all the instructions for what amino acid is supposed to go where. Your RNA writes it down, and then it gets expressed or made in a ribosome. Ribosomes are protein factories. That's why we have to know organelles. And then you have this protein that's the result of a growing polypeptide. Okay, so this is how our different macromolecules connect, which is what I'm trying to really get us to, okay? So when we look at nucleic acids are so important because their DNA and RNA And DNA and RNA get expressed by producing a polypeptide, which turns out to be a protein eventually. Okay? These proteins then help us with our phospholipids. They help us also break down our carbohydrates. So all these things become connected when we start looking at it within the cell part section, okay? Okay, so let me draw out that bond for us as well. What am I doing? Sharing the whiteboard. Okay, so we have our carbon core, as always. Over here, we have our nitrogen, or our amino group, sorry, I would just think of it as nitrogen. It's the amino group with nitrogen. Then over here, we're going to have our carboxyl group. Okay. And then you're going to have some sort of side chain. It can be polar, non-polar, acidic, basic, whatever. There's 20 of them. So another amino acid is coming in based upon what the DNA has said. And things move in a directionality. So if you read it from right to left, right, our nitrogen group our amino group is going to be over here. It's really hard sometimes when you always think of something as like a shortcut. And then our carboxyl group is going to be over here. So the peptide bond is going to be between these two groups, right? This is going to be our resulting peptide bond. And it's between the amino group on one and the carboxyl group on the other. We're going to have our water get removed, right? And then the result will be the peptide bond. So this gets removed via dehydration. And then these guys come together via the peptide bond, and then you have a growing polypeptide. The polypeptide is just the first step onto protein fun. We have to start getting more chemical interactions to get the final protein shape. Because I forgot it, Hannah. There's no R group over here because Aaron forgot it. Okay, let's just go back. So we get this growing polypeptide, but that's just the first step. There's 4 steps that occur to get the resulting complex protein that we really need. And we need it to be the right shape. And the shape correlates with function. And so we're going to have a bunch of steps to go that we'll talk about. But I want to kind of show you a couple of things. So a protein becomes this huge molecule. They're big. They are big macromolecules in the grand scheme of things. So the protein flew from the protein from the flu virus can look like this. So this is one example of what it looks like, and this is kind of that 3D structure. So notice how you have, if you can see, and it depends, like if you can see color contrast well or not. You can see there's 2 different color kind of strings within that. That actually represents 2 different polypeptide chains. And so these polypeptide chains start as a long string, and then they fold in on themselves, and then they oftentimes join with other polypeptides that are folded in on themselves. So these are incredibly complex. So here's the flu, and this is my plug for you to get your flu vaccine, because you can get it free here at Grand Valley. You should all get your flu vaccine. It's not going to give you the flu. There's not a shred of evidence that says that it will give you the flu. Okay? This is a specific shape. The flu vaccine, what's it trying to do? It's trying to flip on your body's response to make the antibody protein. So antibodies are proteins. They're incredibly important proteins. They specifically match the shape of the protein made from the virus. And it has to be perfect because shape and function go hand in hand. There is no exception to that. Asterisk. For us, there's no exception to that. So this antibody protein that your body makes has to match it perfectly. And so notice they do. They fit all happy. You can't see my coloration on that. But then that antibody is going to be able to deal with that protein from the flu virus. The flu virus comes in, right, infects your cell's machinery and starts to make your cells pump out its proteins. Because viruses have part of a nucleic acid in them. They either have part of a DNA or they have part of an RNA. They're using your machinery to then express their genome. And they make you make their proteins, and then your body's like, wait a second, I got to fight this with, I got to make some antibodies that match the proteins. They have to match perfectly. So let's talk a little bit about COVID then, because I think it's well worth kind of looking at. So here's the COVID virus. A couple things to keep in mind now that we have some language down. Lipid membrane. Guess what that lipid membrane is? Oh, it's phospholipids. Guess whose phospholipids it's just like? Oh, ours. Okay? That's the most wonderful thing about living things is that our chemistry is remarkably similar, if not exact, right? We have an envelope protein. That protein is going to be specific amino acids linked together that form a specific shape. We have a membrane protein. Those are going to be specific amino acids that link together to make a specific shape. We have a spike protein. This is how the corona got its name because they look like little crowns. And then inside, not in the nucleus, not in anything, they have RNA. They don't have DNA, they have RNA. So now, when we are trying to think about making a vaccination against, right, what we're doing is basing the vaccination on these proteins and on the proteins that are made from the RNA being expressed by our cell's machinery. So the antibodies have to be made perfectly to match what this RNA is going to get expressed into, because this RNA is the same genetic code that our RNA contains. It's molecularly the same. Okay, so if you had to get corona tested, I had to get one of the random sample ones. They're doing a PCR, and what they're trying to do is amplify this RNA to see if you have some of it in your body. So then, what we're doing now, right, since we have no vaccination to help us with antibody production, we're wearing masks that help to keep us safe because we know that it's a big respiratory transmission. But what else are we doing? We have to wash our hands. This is the link that you could click on if you want. But I wanted to talk about two major things. Okay, soap. Why does soap work so well? Soap works really well because what it does, remember, soap is it hydrophobic or hydrophilic? Or is it maybe both? It's both, right? It's amphiphilic. And so what soap's able to do is it's able to get in that lipid membrane. Very much how soap can help you get butter off your hands if you get squeezed butter all over, right? And so what it does is it breaks apart their lipid membrane and it spills out the contents, and now it's a dead virus, right? Arguably, the virus was never alive, but now the virus can't function. So then, hand sanitizer, which is what this link is kind of getting to, is not all infectious pathogens can be managed with hand sanitizer. So if you get the stomach flu and you throw up, it's not actually the seasonal flu that you're getting the shot for. The seasonal flu that you're getting the shot for is a really yucky respiratory disease that gives you a fever and you feel super junky. Just throwing up is typically the norovirus. Okay, the flu shot does not protect you against that. The norovirus has a really hard shell on it. Hand sanitizer does not kill that. Only soap can get norovirus off of your hands from the scrubbing action. However, with the COVID virus, hand sanitizers work, just like with the seasonal flu. Here's why. It has alcohol in it, okay? It's not as effective as hand washing, but it's working on the lipid bilayer. You have to have at least 62% alcohol. You don't have to have more, but you have to have at least between like 60 and 80 is shown to be the most effective percentage of alcohol, and it has to have time of contact. So if you've been in lab, like in physical lab, you know that you've sprayed your seat before you left, and then you just leave it. The reason why you're leaving it is because that hand sanitizer, the alcohol that has a hydroxyl group on it, needs some time to break apart the lipid membrane. That's what it's working on. And so when you hand sanitize with your hands, on your hands, you need to make sure you have enough that saturates your hands and that your hands are a little wet so that it can work on that lipid membrane. So a lot of the mitigation strategies we're working on aren't actually the product of science, but they're based on these scientific understandings of these chemical properties that we're looking at in our biological setting, okay? So just some food for thought. So let's look at how proteins are made, because viruses make proteins in the same way. So there's 4 levels of protein structure. So we don't just jump to that complex flu protein. We don't just jump to our complex hemoglobin protein. There's multiple steps to get to these complex structures. So notice each one of these little purple dots is an amino acid. And that's not always super apparent as we go. So look here, the book, good or bad, switches between presentations. And so I think just like you're looking at a graph for the first time, you've got to try to decipher, okay, what is this one telling me? So you have the carbon middle, you have your amino group over here, you have a carboxyl group here. Right here, that's a peptide bond. And then you have these R groups. Notice the R groups are kind of all over the place. That's okay. So each one of these purple dots, circles, is an amino acid. So look how many there are. There's oodles. This is the primary structure. So that polypeptide we've been talking about, that's the primary structure. It's the beads on a string, or the pearl necklace, somebody might have said before. So it's just the string of amino acids that have been peptide bonded together. That's the primary level of folding. Oops, sorry, wrong one. There's 3 more to go to get to our very complex protein structures. I'm going to flip through them real quick, and then we'll start there again next week. So you have the secondary structure, which is going to begin to kind of coil or fold it, and it's going to be hydrogen bond interactions. We're going to have the tertiary structure, which is going to be the R groups interacting, and we'll start here again, don't worry. And then that quaternary, or that 4th degree, is when you're going to get multiple polypeptide chains coming together. So collagen that's in your skin and hair, and this is your hemoglobin. That's what helps your red blood cells carry oxygen around. These are big, complex proteins that have multiple polypeptide chains bonded together. So they're incredibly complex. So the chances for things to go wrong is high, and it's amazing that they don't, right? That our DNA gets expressed accurately to reflect the correct amino acid in the correct spot. Okay, so we're going to start there on Tuesday. You have a homework that's going to help you with this. There's lots of stuff in that study area on mastering biology. And remember, Amoeba Sisters are a great supplement to your Pearson resources. They're really simple, really straightforward. It's cute little amoebas, and it's perfect to kind of hear the vocabulary again and again, okay? So stay safe if you're on campus here. Try to figure out how to keep yourself busy. Studying would be one way. Take some walks this weekend. It should be nice out. And I'll see you guys next week. Let me know what questions you have.